Hey science fans, ever wondered how your body knows how to build you? How a single cell becomes a complex organism? The answer lies in the genetic code. This code guides everything from your hair color to your immune system. Imagine DNA as a winding staircase. Each step is made of nucleotide pairs. The sequence of A, T, G, and C forms the genetic code. This code is universal, used by all living organisms.
So, how exactly does the genetic code work? How do those four letters, A, T, G, and C, actually tell our cells what to do? The answer lies in the concept of codons. Think of codons as three-letter words within the genetic language. Each codon corresponds to a specific amino acid. Amino acids are the building blocks of proteins, like individual beads on a string. The sequence of codons in a gene determines the sequence of amino acids in a protein. This sequence, in turn, determines the protein's shape and function. For example, the codon, AUG, not only codes for the amino acid methionine, but also signals the start of protein synthesis. Other codons, like UAA, UAG, and UGA, act as stop signs, signaling the end of the protein chain. The beauty of the genetic code lies in its simplicity and elegance. With only four letters arranged into three-letter words, it's remarkable to think that all the diversity of life we see around us stems from this fundamental code.
One of the most fascinating aspects of the genetic code is its redundancy. What does that mean? Well, most amino acids are encoded by multiple codons. For example, the amino acid leucine can be specified by 6 different codons. This redundancy acts as a safety net, reducing the impact of potential errors during DNA replication or protein synthesis. Imagine you're copying a recipe and you accidentally misspell sugar as sugar. If the recipe still makes sense, you can still bake the cake. In the same way, redundancy in the genetic code provides a buffer against mutations, ensuring that minor changes in the DNA sequence don't always result in a non-functional protein. Even more incredible is the universality of the genetic code. With very few exceptions, the same codons code for the same amino acids in all living organisms, from bacteria to humans. This universality highlights the shared ancestry of all life on Earth, suggesting that we all evolved from a common ancestor billions of years ago. This shared genetic language also has profound implications for fields like biotechnology and genetic engineering. Scientists can now insert genes from one organism into another, and the recipient organism can often successfully read and translate the foreign gene, producing the desired protein. This ability to manipulate the genetic code is revolutionizing medicine, agriculture, and many other industries.
Now, let's zoom in on the molecular machinery that translates the genetic code into proteins. One of the key players in this process is transfer RNA, or tRNA for short. These tiny molecules act as interpreters, linking the language of codons to the language of amino acids. Each tRNA molecule has 2 crucial regions, an anticodon and an amino acid attachment site. The anticodon is a 3-nucleotide sequence that is complementary to a specific codon on the messenger RNA, mRNA. The amino acid attachment site, as the name suggests, is where a specific amino acid is attached. Think of tRNA molecules as tiny trucks, each carrying a specific amino acid cargo to the protein assembly line. The anticodon on each tRNA acts as an address label, ensuring that the correct amino acid is delivered to the right spot on the mRNA template. Ensuring accuracy, aminoacyl tRNA synthetases. But how do the tRNA molecules know which amino acid to pick up? This is where a group of enzymes called aminoacyl tRNA synthetases come into play. These enzymes are the quality control managers of the protein synthesis process, ensuring that the right amino acid is attached to the right tRNA molecule. Each aminoacyl tRNA synthetase is specific to a particular amino acid and its corresponding tRNA. These enzymes have 2 binding sites, one for the amino acid and one for the tRNA. The enzyme brings the 2 together, catalyzing the attachment of the amino acid to the tRNA's amino acid attachment site. This process of attaching the correct amino acid to its tRNA molecule is crucial for the accuracy of protein synthesis. If the wrong amino acid is attached to a tRNA, it will be incorporated into the growing protein chain, potentially leading to a non-functional or even harmful protein.
The protein factories, ribosomes, and their structure. Now, let's talk about the heart of protein synthesis, the ribosome. Ribosomes are complex molecular machines found in all living cells, and their job is to read the mRNA template and assemble amino acids into proteins, following the instructions encoded in the mRNA. Ribosomes are made up of two subunits, a large subunit and a small subunit. The small subunit binds to the mRNA molecule, while the large subunit catalyzes the formation of peptide bonds between amino acids, linking them together into a growing polypeptide chain. The ribosome has three binding sites for tRNA molecules: the A site, aminoacyl site, the P site, peptidyl site, and the E site, exit site. These sites are crucial for the stepwise process of protein synthesis. From code to creation, the stages of translation. Translation is the process where genetic code builds proteins with three main stages: initiation, elongation, and termination. Initiation sets the scene for protein synthesis. The small ribosomal subunit binds to the mRNA at the start codon, usually AUG. Elongation. The protein chain begins to grow. The ribosome catalyzes peptide bond formation between amino acids. Termination. The ribosome encounters a stop codon, signaling the end of protein synthesis. Release factors bind, causing the release of the completed polypeptide chain. The new protein is released into the cell, ready to function.
Proteins are the true workhorses of our cells, carrying out a vast array of functions that keep us alive and thriving. They are involved in almost every aspect of cellular life, from catalyzing metabolic reactions to transporting molecules across cell membranes. Enzymes, a special type of protein, act as catalysts, speeding up chemical reactions within cells. Hormones, another type of protein, act as chemical messengers, coordinating activities between different parts of the body. Antibodies, yet another type of protein, are crucial components of our immune system, defending us against invading pathogens. The diversity of protein shapes and functions is truly remarkable. Some proteins are globular, like tightly wound balls of yarn, while others are fibrous, like long, sturdy ropes. Some proteins are embedded within cell membranes, while others float freely in the cytoplasm. The specific sequence of amino acids in a protein determines its unique three-dimensional shape, which in turn dictates its function. Even a single amino acid change can drastically alter a protein's shape and function, potentially leading to disease. The process of translation, from DNA to RNA to protein, is essential for life. It underpins the diversity of life and our ability to adapt. Different cell types arise from differences in gene expression.
Translation plays a crucial role in evolution. Mutations can lead to changes in proteins. Some changes might be harmful, leading to disease. Others might be beneficial, providing an advantage.
Understanding the intricacies of translation is crucial for advancing our knowledge of life itself. By unraveling the molecular mechanisms of this fundamental process, we gain insights into the origins of life, the evolution of diversity, and the development of diseases. This knowledge empowers us to develop new diagnostic tools for genetic diseases. By analyzing an individual's DNA sequence, we can identify mutations that might increase their risk of developing certain diseases. This information can then be used to tailor personalized treatment plans and lifestyle interventions. Furthermore, a deep understanding of translation is paving the way for innovative therapeutic strategies. For example, scientists are developing drugs that target specific steps in the translation process, aiming to block the production of harmful proteins or enhance the production of beneficial ones. As we delve deeper into the mysteries of the genetic code and the process of translation, we unlock extraordinary possibilities for the future. With this knowledge, we can rewrite the blueprints of life, correct genetic errors, and engineer organisms with enhanced capabilities. Imagine a world where genetic diseases are eradicated before they even manifest. Imagine crops engineered to withstand harsh climates and resist pests, ensuring food security for a growing population. Imagine personalized medicine tailored to an individual's unique genetic makeup, optimizing treatments and minimizing side effects. The future holds immense potential, and our understanding of the genetic code and translation will be the driving force behind these groundbreaking advancements. As we continue to explore the intricacies of this remarkable language, we embark on a journey of discovery that promises to transform our world in ways we can only begin to imagine.